Hello everyone, welcome to HAP201 and welcome to week 1 lesson 1 where we will introduce the reproductive system and begin with a study of the male reproductive anatomy and some physiology. What is the purpose of our reproductive system? Sexual reproduction is present to unite the sex cells or gametes from the male and the female in order to join the chromosomes and create the information needed to start a new life. The gonads are the organs in the female or the male that have two major functions. Number one, to produce the sex cells, and number two, to produce the sex hormones. In the male, the gonads are the testes, and the sex cells they produce, the gametes, are called sperm or spermatocytes. In the female, the gonads are the ovaries, and the gametes are the secondary oocytes. Now, you may have heard in general discussions that the female gamete is the egg. We will not be calling it an egg. If you want to use a proper name, it's the ovum. One is an ovum, multiple are ova, but technically female ovaries do not release ova. Each month, the female releases a secondary oocyte. So this is an immature ovum that is yet to complete the process of cell division called meiosis that will lead to the proper number of chromosomes. We will get into that later. I just want to clarify the proper terminology. The process of uniting the gametes is called fertilization. It takes place in the fallopian tube of the female reproductive tract, and it is where the complement of chromosomes from the sperm cell and the male unite with the chromosomes from the female in the secondary oocyte. After fertilization is initiated, the secondary oocyte completes meiosis and becomes a true ovum, and then the genetic material is united to form a diploid or a cell that has a full complement of chromosomes called a zygote. We'll get into the specifics of this later on. Pregnancy begins when fertilization occurs, but then in order to have a complete pregnancy, the fertilized zygote must then implant into the uterus of the female. That zygote will then undergo several rounds of cell division, and within a few days becomes an embryo. After a few weeks, the embryo develops into a fetus, and after 40 weeks of gestation, in total, we have birth. All right, let's now meet the structures of the male reproductive system. We have already met the testes, also known as the testicles. These are the paired male gonads located in the skin sac called the scrotum. So the testes or testicles are actually housed outside of the abdominal pelvic cavity for a very specific reason, which we will discuss shortly, but keep that in mind. The system of ducts that extend from the testes and help to transport the sperm produced in the testes include the epididymis, the ductus deferens, also called the vas deferens, then the paired ejaculatory ducts, and the single urethra. Now, the urethra in the male is special because it begins at the base of the urinary bladder, passes through the prostate gland, and then through the length of the penis. The urethra in the male is a common organ or tract for both the urinary system, allowing for the excretion of urine, as well as the reproductive tract, allowing for the ejaculation of semen. And semen is a mixture of sperm and the secretions of the reproductive accessory glands. So these glands include the seminal vesicles, the single prostate, and the paired bulbourethral glands, also known as the Cowper's glands, spelled just as it sounds, Cowper's. All right. The supporting structures include the scrotum, which houses, again, the testes, and the penis. So the penis is the organ of intercourse, the organ of copulation. The purpose of the penis is to support the urethra and deliver the sperm into the female reproductive tract. Here we have a sagittal section through the male pelvic region, which shows us a lateral view of the reproductive as well as urinary structures. So you'll notice that the urinary bladder is a large sac that stores urine, and it sits superior to the prostate. So the urinary bladder is part of the urinary system, but the prostate is part of the reproductive system. The urethra begins at the base of the urinary bladder and then passes through the prostate, through the pelvic floor, and through the length of the penis. The urethra then transports urine during urination, but also transports sperm mixed with secretions to make semen during ejaculation. So the urethra is common to both the urinary and reproductive tracts in the male. And because of this, these structures are often studied together as a urogenital or genitourinary system in the male. Let's look at the other structures of the male reproductive system, beginning with the testes. So a single one is called a testis or a testicle, and it is the egg-shaped structure about the size of a walnut located in the scrotum. So scrotum is skin that extends from the pelvic floor and surrounds and houses the testicles as well as an epididymis. The epididymis is the beginning of the duct system for the male reproductive tract. The epididymis has three main functions. Number one, it is where sperm are stored and wait to be ejaculated. Number two, the epididymis is a site of sperm maturation. So sperm that are produced initially in the testes are not capable of fertilization. They require further maturation by exposure to secretions from the epididymis. And even then, the sperm is not capable of fertilization, but we'll discuss that later. But the sperm undergo maturation in the epididymis. And finally, sperm that are not ejaculated and become senescent or old, worn out, damaged, will not kill a male, but they will be broken down or degenerated in the epididymis, and the components will be recycled. All right, if sperm is ejaculated, it will leave the epididymis and then travel the length of the ductus deferens or vas deferens. So that is a large tube or duct that you see extending from the tail of the epididymis inferiorly in the scrotum and extends up through an opening into the pelvic region. It moves over anteriorly the pubic bone at the pubic symphysis, then passes laterally on either side of the urinary bladder, and then finally ends on the posterior aspect of the urinary bladder in an opening called the ampulla, or widening rather, of the vas deferens, called the ampulla of the ductus deferens. The ampulla of the ductus deferens or vas deferens converges or merges with the duct from the paired seminal vesicles. The seminal vesicles are one of the glands, and they lie posterior to the bladder. Together, the duct from the seminal vesicle joins with the ampulla of the vas deferens to form the ejaculatory duct, and the ejaculatory duct passes through the prostate gland. The prostate gland is a site where the ejaculatory duct now converges with the urethra, which began at the base of the urinary bladder. The part of the urethra that passes through the prostate is called the prostatic urethra. The shortest segment of the urethra extends from the prostate through the urogenital floor or the muscular floor as the intermediate urethra, also called the membranous urethra. And the longest part of the urethra passes through the length of the penis all the way to its distal tip or opening called the external urethral orifice. So the external urethral orifice is where urine is excreted from the body. It's also where semen is ejaculated from the body. The penis itself begins at the base of the pelvis as the root. You can see a part of it laterally called the bulb of the penis. The bulb is formed by the more medial column of spongy tissue in the penis called the corpus spongiosum. The spongy columns or tissue in the penis have many cavities, so they have the appearance of sponge, but they also become filled with blood that causes engorgement and stiffening of the penis, leading to an erection. So the corpus spongiosum is the more medial column. It surrounds and supports the urethra. Lateral and slightly ventral to the corpus spongiosum are two columns of spongy tissue called the corpus cavernosum. So one is a corpus cavernosum. We'll see the plural later on. So together, these three columns of erectile or spongy tissue allow the male to experience an erection, which is necessary for the delivery of sperm into the female reproductive tract. Okay. Posterior to these reproductive structures, we see the rectum, which is the distal part of the digestive tract. But you'll notice that in this entire perineal region, the male has two orifices or opening. For the digestive tract, it ends at the anus. And for the reproductive and urinary tracts, the single opening is the external urethral orifice. All right, I hope we covered everything there, but this is an important diagram to study and understand. On this slide, we will consider the scrotum and associated structures. We mentioned that the scrotum is skin that houses and surrounds the testes. Deep to the skin itself, there is a hypodermis layer, and the scrotum is also divided into a left and right half by a band of connective tissue. Internally, it forms the septum, and superficially, just beneath the skin of the scrotum, it appears as a scrotal raphe, very pronounced ridge that runs right down the center of the scrotum. Now, you may wonder, why are the precious testicles housed outside the body? That is because the production of sperm, a process called spermatogenesis, is optimal at 2 to 3 degrees below body temperature. And this is why the testes are housed outside of the body in the scrotum. Now, during gestational development, the testes, like the ovaries in the female, develop within the abdominal pelvic cavity, but at about 7 months, the testes usually descend through a passageway called the inguinal canal into the scrotum, preparing the testes for sperm production later in life. Deep to the skin of the scrotum, there are two layers of muscle, the cremaster and the dartos muscles. Together, they work to regulate the temperature of the testes for spermatogenesis. So one contracts to elevate the scrotum and bring the testes closer when it's cold outside, or that muscle will relax to move the testes further away for cooling. Another layer wrinkles the scrotal skin to retain heat when it's cold, or again, relaxes to dissipate heat when the body is warmer. So they work together to again maintain the optimal temperature of the testes for spermatogenesis. Inside the scrotum, there are several structures associated with the testes, including blood vessels, lymph vessels, nerves, and the ductus deferens, which are bundled together in a structure called the spermatic cord. And the spermatic cord extends from the scrotum through the opening called the inguinal canal into the pelvic cavity. Let's look at them on the diagram on the next slide. So this is an anterior view of a frontal section through the scrotum. Ouch! All right, what do we have here? The penis has been cut to show the two columns of spongy tissue making up the corpora cavernosa, that's the plural for the two, and then the more medial and dorsal corpus spongiosum of the penis. This diagram also removes part of the penis to show the structures of the scrotum. So here you can see the distinct scrotal septum that separates the scrotum into the right and left half. In each half, there is a testicle. The testicles are surrounded by the cremaster muscles. These are the ones that elevate or lower the testicles. And we also have the dartos muscle that is more associated with the skin of the scrotum, wrinkling it to, again, retain heat or relaxing to dissipate heat. The testicles are each surrounded by two layers of connective tissue. There is the more superficial tunica vaginalis, and deep to it, we have the tunica albuginea. The tunica albuginea, as you'll see on the following slide, extends deep into each testis to separate it into partitions where the sperm are produced. On this diagram, you can also see the superficial ridge that's created by the scrotal septum called the raphe. And we also have the structures that make up the spermatic cord, which include the lymphatic vesicles and your blood vessels. The testes are highly vascularized because, again, they produce hormones, which have to be transported via the blood out of the testes into the general circulation. We also need blood supply for engorgement of the entire area during sexual arousal. And of course, the nerves that signal the various changes, including contraction of the muscles that make up the ductus deferens during ejaculation. So more superiorly on the right, you can see the label for the inguinal canal. That's the passageway from the scrotum into the pelvic cavity. And we also have several ligaments supporting these structures. Here we have the testes or testicles, which again are the gonads of the male reproductive system. The testes contain several hundred seminiferous tubules, which are the site of sperm production during spermatogenesis. The tubules surround a lumen, which will eventually collect the sperm, but the cells that give rise to the sperm are called spermatogenic cells. So these make up the walls of the seminiferous tubules, and the spermatogenic cells are at different stages of maturity. The more immature cells are located superficially, and the more mature cells, as become sperm, of course, are closer to the lumen. Sperm production begins at puberty with a rise in the levels of the hormones that stimulate puberty, and specifically spermatogenesis. Within the seminiferous tubules, there are also Sertoli or sustenacular cells or nurse cells that surround the spermatogenic cells. And during the process of spermatogenesis, these Sertoli cells play several important roles. For example, they nourish and control the movement of developing sperm on their way to being released into the lumen. Another important function is that they form the blood testis barrier. And in this way, the cells form tight junctions that block immune cell entry and recognition and eventual destruction of the developing spermatogenic cells. So the spermatogenic cells are blocked from immune cell exposure. Therefore, these cells can develop into mature sperm cells without being destroyed by the immune system that would recognize these cells as foreign because they were not present during infancy and early childhood when the immune system was being programmed to recognize self versus non-self cells. The Sertoli cells produce several secretions. For example, they produce the hormone inhibin, which rises as sperm cell numbers and the rate of spermatogenesis increases. Inhibin, as you will see, feeds back to the brain to suppress the hormones that stimulate spermatogenesis. And in this way, spermatogenesis is controlled in a negative feedback manner. There are several other functions of the Sertoli cells, which are summarized in the document Cells of the Testes. To mention them quickly, the Sertoli cells also phagocytize excess cytoplasm from the developing sperm cells, particularly towards the end of the spermatogenesis process. That last step is called Spermiogenesis, where the cells take on the appearance or morphology of sperm, which requires removal of excess cytoplasm. The Sertoli cells also produce the fluids that are necessary for the protection and transport of the sperm. The Sertoli cells secrete a protein called androgen binding protein that binds testosterone and keeps testosterone levels high around the spermatogenic cells during spermatogenesis. And the cells also bind to and respond to the effects of follicle stimulating hormone from the anterior pituitary, in that FSH supports spermatogenesis along with testosterone. The other major cell types that are found in the testes are the Leydig or interstitial cells, and these are located between the seminiferous tubules. These are the cells that bind luteinizing hormone from the brain, the anterior pituitary, and lead to the production of the male sex hormones, testosterone being the most common form. All right, so here we have a sagittal section and a lateral view of a testicle. You can see that's associated with the structures that make up the spermatic cord, including the vas deferens that extends from the epididymis, which is a continuation really of the testes. So let's start with the seminiferous tubules of the testes where sperm are produced. The seminiferous tubules are collected into segments called lobules. Each lobule has 1 to 3 seminiferous tubules, and the lobules are defined by the invagination of the tunica albuginea. So that was one of the connective tissue layers around the testes that extends internally to form the septa that divide the testicle into lobules, each lobule containing a few seminiferous tubules. Once sperm enter the lumen of the seminiferous tubules, the movement of the Sertoli cells, along with fluid pressure, move the immature sperm cells into the rete testis of each testicle, and from there they are pushed into the epididymis. The epididymis has 3 regions. The head of the epididymis is the proximal region, which is continuous with the rete testes. From there, sperm move into the body of the epididymis and eventually into the tail. While the sperm are stored in the epididymis, they undergo maturation or degeneration and recycling of the components. Also note the blood vessels, lymphatics, and nerves that are associated with the testes and the epididymis, both housed within the scrotum, of course. And all these structures are bundled together in the spermatic cord that take these structures into or out of the pelvic cavity, including the vas deferens, which is a continuation of the tail of the epididymis and is the tube that brings the sperm from the scrotum into the pelvic cavity. Here we have a cadaver view of the testes. On the left, we have the more superficial structures. So you can see the tunica vaginalis, which is more superficial to the tunica albuginea, which extends deep into each testicle to form the septum and define the lobules. In this diagram, we also see the head, body, and tail of the epididymis. And on the right, you can see roughly the seminiferous tubules, the rete testis, the tunica albuginea that form the septum and lobules, as well as the more superficial tunica vaginalis. In this transverse plane through a testicle, you can see the Reedy's testes and lobules well defined. You'll notice that within each tubule, there are several spermatogenic cells in different stages of maturation. So the more immature spermatogonia are located superficially. They develop into the primary spermatocytes, and then the secondary spermatocytes, spermatids, and eventually spermatocytes or sperm cells. And as the cells are dividing and maturing, they are being pushed further toward the lumen of each seminiferous tubule. The basement membrane surrounds each seminiferous tubule, and superficial to that, between the seminiferous tubules would be the interstitial cells that produce testosterone under the influence of luteinizing hormone. Here we have a transverse section of a part of a seminiferous tubule. So we are looking at the cells within a region of the tubule, in addition to a few interstitial cells outside of the tubules, the extracellular components, blood capillaries, etc. We also have the defining and confining basement membrane that surrounds the seminiferous tubules. Within the tubules, we have the developing spermatogenic cells as listed on the right, and these are surrounded by the testicular or supporting cells, the Sertoli cells. So these are the ones that nourish the developing spermatogenic cells. They provide the fluids that support and transport the sperm cells. Here they form tight junctions that make up the blood testes barrier, etc. So the spermatogenic cells are listed in order of increasing maturity. Males are born with several spermatogonia. A single one is a spermatogonium. And these cells are located on the outer or superficial area of the seminiferous tubules. They are diploid, as denoted by the 2N designation, meaning that they have the full complement of chromosomes, or 46, which are organized into 23 pairs. Hope that sounds familiar. Spermatogonia undergo mitosis starting at around puberty. So the process of spermatogenesis is initiated at around the age of puberty due to rising levels of sex-related hormones. When the spermatogonia divide into two daughter cells, one becomes a primary spermatocyte and one becomes a new spermatogonium that stays near the basement membrane. This means that males have a continuous and unending supply of progenitor cells to make sperm. Males can make sperm until they die, and they can father children way up into their older adult years. The quality of the sperm is questionable, but they can still produce sperm. Now, the primary spermatocytes will then undergo meiosis 1. So in meiosis 1, the number of chromosomes is doubled, and the cells separate into two daughter cells, each having half the number of chromosomes, or 23, making them haploid, as denoted by the single N. But recall that during meiosis 1, there's a double duplication of sorts, where each chromosome actually has two sister chromatids, each carrying a copy of the genes for that chromosome. So a primary spermatocyte undergoes meiosis 1 to become two secondary spermatocytes, and the secondary spermatocytes undergo meiosis 2, where the sister chromatids in each chromosome are separated into individual cells so that each secondary spermatocyte forms two haploid spermatid, and overall each primary spermatocyte gives rise to 4 equally viable spermatid. Spermatid have the correct number of chromosomes with respect to gamete formation. However, they do not have the structures and the morphology necessary for fertilization. So spermatid undergo further differentiation to become sperm cells. There are no cell divisions, they just change their shape. So this involves the absorption and phagocytizing of the excess cytoplasm by the surrounding Sertoli cells that we described on the previous slide. The spermatid acquire a tail and a midpiece and a head, which we will describe on a future slide. But these are the structures that are found in a mature sperm cell, also known as a spermatocyte. On this slide, we will review the steps of spermatogenesis as they occur in the seminiferous tubules. The stem cells, the precursor cells, the spermatogonia begin mitosis at around age 10, 11, 12, puberty in the male. As these cells undergo mitosis cell division, one of the two daughter cells remain near the basement membrane as a stem cell to provide a constant supply of spermatogonia. The other cell is pushed between the Sertoli cells and develops into a primary spermatocyte. It is still diploid. It has 2 copies of each chromosome, or a full complement of 46. During meiosis, a primary spermatocyte divides into 2 secondary spermatocytes, not spermatid, as you might have seen on the original slide. These spermatocytes are haploid, meaning they have half the number of chromosomes. However, within each cell, each chromosome has 2 sister chromatids, okay, of each pair of chromosomes. Then finally, the secondary spermatocytes undergo meiosis too and become spermatids. So in the end, there are 4 spermatids formed from each primary spermatocyte. These are truly haploid, having only 23 chromosomes, or half the full complement. The process is summarized here again in this diagram, where the spermatogonium undergo mitosis to form 2 diploid cells, one staying near the basement membrane as a stem cell, the other going on to become a primary spermatocyte. The primary spermatocyte undergoes DNA replication, and this is the step where some variation in the genes can occur as the paired chromosomes align together and can actually exchange pieces so that genes that were initially on one of the pair of chromosomes are exchanged with the genes on the other form of that same chromosome. We'll talk about that later. Now, during this step of meiosis 1, each primary spermatocyte divides into 2 haploid secondary spermatocytes. So each has 23 chromosomes, but each chromosome has 2 copies of the DNA as chromatids. Then the secondary spermatocytes divide into 2 spermatids each, so you get a full 4 from each primary spermatocyte. Each spermatid has a single copy of each chromosome, but half the number of the total that we began with. So each spermatid has 23 chromosomes. These spermatid then undergo maturation to become sperm cells, and that process of maturation, differentiation, is called spermiogenesis. And as these cells are dividing and maturing, again, they're being pushed toward the lumen of the seminiferous tubule. All right, the last stage of spermatogenesis is spermiogenesis, where the spermatid mature into spermatocytes or sperm cells. This step is influenced by testosterone. So testosterone is absolutely necessary for adequate, proper spermatogenesis. Testosterone binds directly to the Sertoli cells that surround the developing sperm cells, and it also binds to a protein produced by the Sertoli cells, which is called androgen binding protein. So the ABP helps to keep testosterone levels high around the developing sperm cells. As the spermatid mature into sperm cells, they acquire these unique structures, including an acrosome, which is a cap that surrounds the head of the sperm cell. The tail is called a flagellum. That is required for the motility of the sperm cell through the female reproductive tract. The spermatid, on their way to becoming sperm cells, shed excess cytoplasm, which again is phagocytized by the surrounding Sertoli cells. And because these sperm cells have a mighty long way to go through the female reproductive tract, they also have an increase in the number of mitochondria, which will produce ATP to power the flagellum. So the elongated sperm cells, once mature, are released into the lumen of the seminiferous tubules, and then as they are suspended in the fluids created by the Sertoli cells, they will be pushed towards the ducts of the testes, the Redes testes, and eventually into the epididymis. So here's a number for you. Males of reproductive age produce on average 300 million sperm per day. Now, even though that's the number of sperm produced, note that the steps of spermatogenesis take several days to complete. So any one sperm requires up to 2 months to become mature. Okay? Now, once sperm are formed, they will remain in the epididymis until they are released from the body during the process of ejaculation. Once ejaculated, sperm can actually survive 48 hours in the female reproductive tract. Keep those numbers in mind. The mature sperm has these regions. We mentioned the head. This is the part of the cell that contains the nucleus, as well, and most importantly, the haploid DNA, or the 23 chromosomes. Surrounding the head, protecting it, is the acrosome, and this vesicle structure also contains enzymes that break down and penetrate the protective layers around the secondary oocyte, and these enzymes are necessary to access the membrane of the secondary oocyte for fertilization. The middle piece is the motor that contains the mitochondria and the proteins that power the tail or flagellum for locomotion. So the mitochondria again produce the ATP. The tail actually creates the motion that allows for the motility of the sperm through the female reproductive tract on the way to encounter a secondary oocyte. Here we have a single sperm cell or spermatocyte. Definitely does not look like the original spermatogonium or even the spermatid that preceded it. The mature sperm has several distinct structures divided into the head region and the tail region. The head includes the acrosome that contains the enzymes that break down the protective layers around the secondary oocyte in the process of fertilization. We also have the region that contains the nucleus and the 23 chromosomes. It is attached to the tail by a neck region. The tail has the middle piece that contains several mitochondria and other structures that power the flagellum, which is the tail whipping part that moves the sperm through the female reproductive tract. All right, let's now consider the hormones that are responsible for spermatogenesis, as well as the development and features of male reproductive structures. At puberty, levels of gonadotropin-releasing hormone, or GnRH, from the hypothalamus peak, and these high levels of GnRH stimulate the anterior pituitary gland to release two gonadotropin hormones. They are luteinizing hormone, or LH, and follicle-stimulating hormone, FSH. Luteinizing hormone binds to the Leydig or interstitial cells within the testes outside of the seminiferous tubules and stimulate these Leydig cells to secrete androgens, the most dominant form being testosterone. So your androgens are your masculinizing hormones. Follicle-stimulating hormone binds to Sertoli cells along with testosterone, and together FSH and testosterone stimulate the Sertoli cells to secrete that androgen binding protein we mentioned. And ABP help to keep testosterone levels high in the seminiferous tubules because, as their name suggests, they bind to testosterone. Testosterone, again, is required for the completion of spermatogenesis. Now, as the rate of sperm cell production increases, the Sertoli cells release the hormone inhibin. It feeds back to the anterior pituitary to suppress FSH, and in this way it helps to regulate the rate of spermatogenesis in a negative feedback regulation mechanism. So inhibin from the Sertoli cells feeds back to suppress FSH, which stimulates the Sertoli cells, while rising levels of testosterone from the Leydig cells feed back to suppress LH and GnRH to decrease the production of testosterone. This slide is an excellent visual presentation of the hormonal pathways that control the testes and spermatogenesis. So again, gonadotropin-releasing hormone from the hypothalamus peaks at around puberty and targets the anterior pituitary to release both FSH and LH. Let's follow LH first. LH then targets the Leydig or interstitial cells of the testes that surround the seminiferous tubules, and there LH stimulates the Leydig cells to produce testosterone. Rising levels of testosterone then feed back to the hypothalamus to suppress GnRH, as well as to the anterior pituitary to suppress LH secretion, and in this way, testosterone regulates its own levels through negative feedback. On the left of the diagram, we see that GnRH also stimulates the release of FSH from the anterior pituitary. FSH targets the Sertoli cells, those sustentacular or nurse cells that surround the developing spermatogenic cells. In response to FSH and testosterone, the Sertoli cells will produce androgen-binding protein that binds to testosterone and keeps the levels of testosterone high around the developing spermatogenic cells, allowing testosterone to support the final stages of spermatogenesis. Rising levels of sperm cells causes the Sertoli cells to release inhibin hormone, which feeds back to suppress FSH, and in this way, the rate of spermatogenesis will decline. But decreasing levels of spermatogenesis will then lead to less inhibin, which then feeds back to allow for more FSH, which will stimulate spermatogenesis. So in this way, testosterone and FSH regulate spermatogenesis in a negative feedback manner. Now, testosterone is not only necessary and responsible for spermatogenesis, testosterone has several effects throughout the body. In certain tissues, testosterone is converted into another androgen called DHT, which stands for dihydrotestosterone. Both testosterone and DHT bind to the same receptors and have the same effects. These include the development of a male during embryonic development. Now, you may wonder, where does that testosterone come from? Before the testes develop, males and females produce testosterone and other androgens in the cortex of their adrenal glands. But the rising levels of these androgens lead to the male pattern of development in male fetuses, and that involves signals from genes on the Y chromosome of the male. We'll look at that later. Once delivered or born, the androgens, testosterone, DHT, and other forms, are responsible for the enlargement of the male sexual organs, and later on the expression of male secondary characteristics which begin at puberty. So what does this include? This includes the development of hair in a male pattern, so the development of facial hair, chest hair, back hair, pubic hair, axillary hair, hair everywhere, mustache, everything, right? We also have enlargement of the reproductive organs, penis, the testes, the scrotum. The Males' vocal cords lengthen, causing a deepening of the voice. Testosterone, as you recall from first semester, is also responsible for the growth spurt that males experience during puberty and adolescence. And it's also responsible for the increased sex drive or libido in males. So the anabolic effect and protein synthesis stimulated by testosterone is responsible for growth of muscle and bones that lead to the growth spurt. And testosterone has several other receptors in the body that lead to the development of these secondary male characteristics. This slide just reviews what we talked about with respect to the regulation of testosterone production through gonadotropin releasing hormone, LH, the interstitial cells that produce the testosterone, and then the negative feedback or suppressive effect of rising levels of testosterone on both GnRH and LH. All right, this slide just summarizes what we said earlier about the effects of testosterone in the male. Again, testosterone is converted into dihydrotestosterone in several target cells, such as the prostate gland, within the seminiferous tubules, etc. They both bind to the same receptors internally within the nuclei and stimulate transcription of genes that lead to these testosterone-related effects. So again, testosterone is necessary for the prenatal development of male organs. It is also responsible for the descent of the testes down into the scrotum at around 7 months. Very important. Testosterone stimulates the development of the male sexual characteristics, broadening of the chest, hair, deepening of the voice, the growth spurt, the anabolic effect, and increased libido.